Welcome to this online lecture in the Advanced Earth Observation course on remote sensing and marine applications at Wageningen University. So naturally, there are many use cases of Earth observation data in the marine sciences. There's harmful algal bloom mapping, coral reef health monitoring, sea surface temperature estimation, oil spill detection, sea ice monitoring, or marine debris detection. And each of these deserve their own lecture on their own. But I will basically focus on these two topics, harmful algal blooms and marine debris detection, because they become a fairly essential part of understanding how we can use remote sensing data in this marine science field, and a lot of ideas will translate to other disciplines. So for each of them, I will go through a first part on the problem setup and then talk about the role of Earth observation. And I will basically leave notes in the sections below so you can skip ahead and you don't have to watch the entire lecture to get to individual parts. So let's start with the harmful algal bloom mapping. So what are algal blooms in general? So they are rapid growth of algae and cyanobacteria of various types. So I have put some images here of different types of algae that can cause algal blooms. And algae is more like an informal term for a large and diverse group of photosynthetic eukaryotic organisms. The core part here is they all perform, or most of them perform, photosynthesis. And cyanobacteria are also almost found everywhere, and the harmful algal bloom term is more the scientific term, but other terms like red tide are also used, but not all red tides are red in general. So why are they harmful in the first place? So they deposit basically bad smelling clumps in the dead cells in the water, and that impedes recreational uses. So you know basically these types of images where the water is very green, you know these types of like warning signs, but this is like not good for the recreational uses, but even worse, if you use some cyanobacteria, they produce toxins and that can cause like rapid death of like animals in the environment. So cyanobacteria dominance is a major risk for human and ecosystem health, and monitoring that with remote sensing is a big part in established monitoring systems. So what causes algal blooms? So first one is warm temperatures. So we have here two comparisons of good algae and bad algae. Good algae like diatoms, they like basically not particularly warm water. So if you look at here 20 degrees to 25 degrees, all the diatoms are still making a large fraction of this biomass here as it is on the y-axis, while the temperature is on the x-axis. But when the temperature rises above 25 degrees, cyanobacteria will basically flourish, and they like it more in these warmer waters, and they will take over the population above 30 degrees, which is naturally a problem with like warming sea temperatures, with climate change, that algal blooms are getting more frequent, and especially algal blooms regarding cyanobacteria are a threat because they produce toxins. A second factor is nitrogen and phosphorus, which is an important field in areas where there's heavy agricultural use, for like in the Netherlands, where fertilizers are washed into the waters and then end up in the oceans, and they increase the total phosphorus content here in microgram per liter. And on the y-axis, you see the biomass of cyanobacteria, and the two lines are the trend in cyanobacteria and the risk of having a cyanobacterial takeover. And what you see is basically if phosphorus increases or nitrogen increases in the water, then the risk of the takeover basically increases, and those cyanobacteria cause a bloom and push away the good algae like diatoms. Another factor that you might not think about too much is no wind, because cyanos and diatoms have a different mass, and compared to water, cyanos will float on the top and diatoms sink on the bottom, and without wind there is no natural mix in the water column, and so they will separate out and they will cause like images like this, where you have floating algae on the top and they can accumulate and then they produce these toxins. And naturally there is like global programs available, as harmful algal bloom mapping is a fairly established field. So on a global level you have here the UNESCO initiative that agglomerates data from national reports, regional monitoring networks, scientific publications and citizen science contributions, and also satellite remote sensing. On a more local scale, you have here the US National Office for Harmful Algal Blooms that keeps a track of all the algal blooms that happened in the past and also is able to do forecasting of algal blooms in the future. And finally, on the European side, you have, for instance, this in ITERAC project S3 EuroHub that monitored the English Channel between England and France for harmful algal blooms with a Sentinel-3 sensor. On a more national scale, there's also citizen science projects, like here this app developed at Wageningen University, where everybody can report sightings of harmful algal blooms in like inland water bodies to a common server, and that information then can be used to catalog existing algal blooms, but also forecast future algal blooms given these factors like wind, temperature. So some takeaways here. So algae is a fairly diverse group of microorganisms, and most of them perform photosynthesis, which will be quite relevant for identifying them with remote sensing. Factors causing blooms: high temperature, high nitrogen, phosphorus content, and no wind. And there's established monitoring platforms available on a global scale, but also on a national scale, and also citizen science projects here in the Netherlands. So let's go to the second part of like the role of Earth observation in harmful algal blooms, and how can we use the properties of algae basically to identify them in satellite images. So first of all, how can remote sensing and Earth observation help? So we can find algal blooms in general relatively easily, so they are detectable, but most existing methods, they produce false positives. So not all flagged pixels are necessarily algal blooms. And most sensors that we have a limitation trade-off between spatial resolution in terms of pixel size, spectral resolution in terms of number of bands, and temporal resolution in terms of number of acquisitions, because we also would like to have a frequent revisit rate. So on the high resolution set, we have classically Sentinel-2, Landsat 7, and Landsat 8. With like these spectral bands over the general windows where we can see something through the Earth atmosphere, where we have atmospheric transmission, and we see that most of those bands naturally align. And generally we have fewer bands when we have a higher resolution, because there's less light that can hit the sensor over a smaller area. So these bands basically are our sensors that we can use to measure the reflectance at these specific wavelengths when we know the spectra of the algae. On the lower resolution side, we have a much more fine-grained picture of different spectral bands, especially in the near-infrared range, which will be quite relevant for different photosynthetic pigments. And we have the MODIS satellite and a Sentinel-3 satellite that produce images at a fairly high temporal resolution of 1 to 2 days, and that we can basically use to make a large-scale analysis of pixels of several hundred meters over, like, let's say, the English Channel, as in this Interact project. So here is an overview of existing sensors. So we have the low-resolution sensors here on the top. MARIS, Sentinel-3, MODIS, they have a fairly high temporal revisit period, and they have also several bands that are relevant for the mapping of harmful algal blooms. On the high resolution side, we have the Landsat series and the Sentinels that take like 20 to 30 meter pixels, where also the Sentinels have a fairly high temporal resolution and moderate coverage in terms of like spectral bands. So let's look at the satellite imagery specifically. So here we have two Landsat images of two severe algal blooms in Lake Erie in 2015 and 2011. And even though both are severe blooms, they have a very different appearance in true color, so in RGB. And that's a challenge because every algal bloom is different in terms of composition and what causes it. And that has effect in its reflectance, and that's what causes this different appearance. So while the left one is fairly clearly green, which we would associate with like algae in general, the right one is more white, which can also be sediments. But it's not that easy, and naturally we need more spectral bands to distinguish different types of algae. And there's also many shades of different algal blooms in general. So I took up this paper here that is a perfect title on many shades of red tides, where red tides is also a different term for harmful algal blooms, where they compared 27 different species on 100 images, and they looked at the spectra of the different species of algae causing algal blooms and compared them with like how they appear visually. So I can give you a few examples just to highlight the diversity of algal blooms in our aquatic environment, in this case in the open oceans. So here we have like an algae that is responsible for a red color, which gives red tides its name, but not all red tides are red. And what we see here is on the top, we have the RGB in true color, and here we have a false color composite with a red edge, green and blue. And we see in the false color composite, we see a fairly clear red line here that is not super visible in the RGB. And this red line makes perfect use of this false color composite, because when we look at the spectra, we see a very characteristic spike here in the 700 nanometer range, which is basically just the red edge band that Sentinel captures. So this is like a perfect visualization here, specifically tuned probably for this like one algae. But this false color visualization and this spectral profile can be very different for different types of harmful algal bloom causing species. So here's another example that looks very different, which is basically these dinoflagellates when a true color and the same false color composite, which does not make that much of a difference anymore because the spectra is fairly flat, so there is not a specific spike. So it's rather reflectance across the entire visible range and also the near infrared. That's why these lines in those images, they appear more white. But you have many shades of algal blooms. So here, for instance, is another species that has two very characteristic spikes, which makes it look a bit more yellow, especially in the false color composite. The true color is still a bit difficult to analyze, but the main takeaway here is that there's a large heterogeneity in the different types of algal blooms. So, takeaway of this, there's a need for a custom design of spectral indices depending on the type of harmful algal bloom that you're interested in and on the sensor that you have available here. So here's all the spectra together with some associated images, and you see that there is many different types of harmful algal blooms that are interesting to analyze. So let's look a bit into the underlying processes in the algae that cause these spectra and look at the differences, because it's much more heterogeneous as in the land field. So typically we have some photosynthetically active pigments like the chlorophylls, chlorophyll A and chlorophyll B, that almost all land plants use, but not all algae use both of these pigments. Also, you may know beta-carotene that is commonly used in land plants as well, but there's also other photosynthetic pigments that are specific to cyanobacteria, for instance. So the different spectra are basically caused by the different biology of those algae. So what we know from like land vegetation does not really translate to the marine field. So let's have a small recap first on some of those examples. And typically in land vegetation, you have a very characteristic spectrum like this one, where you have a high absorbance in the blue and in the red and a high reflectance in the green, which makes the vegetation green. And then you have this massive spike in the near infrared that is very characteristic for land vegetation. But the core part here is while this absorbance here is caused by the chlorophylls, this reflectance is caused by the scattering of near infrared light in the leaf. And that causes basically this high spike. But this is unique to this leaf structure that we have in a lot of terrestrial plants, which is not necessarily the case on the water. So we have this typical red edge. But note, there's also leaves on the water. So there's, for instance, this macroalgae called sargassum that forms leaves and these large carpets. And you can see a classical red edge here on the sargassum patches. So some of the idea still translates, but when we go to, for instance, cyanobacteria, you see that you have these individual cells here. There is an absorbance here on the high reflectance in the green, high absorbance in the blue and in the red, but there's no red edge at all because there's no leaf structure. Do you know why? Because there's no leaf structure in the plant, so this is why we don't have a red edge effect here. So let's look at cyanobacteria in particular as an example of how to go deep into the analysis of different algae species specifically. So we'll focus here on these cyanobacteria, and we will see how we can design spectral indices specifically to analyze these. And in particular, we're interested in the question of like different absorbance in chlorophyll. And the main question I want to pose here is does cyanobacteria use chlorophyll A and chlorophyll B or only one of the two? Also, does it use chlorophyll A or chlorophyll B? And you can pause the video and you can try to figure it out given the spectrum, but I will just go ahead for now. And what we see here is that there is a missing absorption spectrum in the blue side. So it rules out basically chlorophyll B. And here is the good absorption spectrum in the chlorophyll A side, where you see that also the reflectance goes down where the chlorophyll A absorbs. So, but you may also say, okay, what about this step here? That was very much on the chlorophyll B absorbance spectrum. That is actually caused by phycocyanin, which are these small antennae on top of the photosystem too in the cyanobacteria. So there's different pigments in cyanobacteria that absorb light, causes a specific reflectance that is unique to cyanobacteria or to any species that use this photocyanin as a pigment. So this level of detail is useful and helpful if you want to design spectral indices specifically for algal blooms, that you go deep and you understand what is like the underlying physical properties and the objects that you are measuring. And naturally, we have some tools to design indices or families of indices for specific cases. And these families, so there's two main families. One is the normalized difference indexes that, like NDVI, are fairly commonly used in land vegetation. And also there are spectral shape indices that we'll talk about a bit later. But this first one should look fairly familiar from the land vegetation case, where in NDVI you really want to measure this like spike or this increase in near-infrared reflectance by doing this ratio between the two bands and the subtraction, so you can measure basically the slope of this line. And that has been also used in cyanobacteria, but not in the same spectral range, but basically in this absorbance range. So you can measure basically how sensitive is this range to the chlorophyll A in this like cyanobacteria. So the same idea of normalized difference indices can be translated from land vegetation to cyanobacteria, but you will need to use different bands. And which bands to use is a matter of like how well you understand the individual algae. So here is some proposed indices, and there's plenty more in the literature depending on the type of algae and chlorophyll you're interested in. And there's also a matter of analyzing and visualizing the images accordingly with what you would like to identify. So let's go a bit deeper in this like normalized difference chlorophyll index and try to get a geometric intuition what this ratio does. So you have basically the subtraction over the addition. And what we want to do mainly is to measure like the slope between these 2 points on this spectra that we are interested in, so Anabaeana, against the spectrum of water, which is usually the background that we would like to separate the Anabaeana on. So what we can do here is then basically add one side on top of each other and subtract it from the other. And what we get by doing this is basically a slope. So we can divide these two, and then we can estimate a slope between these 2 points. And when the slope is positive, then the index will be positive. When the slope is negative, the index will be negative. In that case, you will be able to fairly easily separate this chlorophyll content with the water background by seeing if this index is positive or negative, or if the slope is positive or negative. Another family of indices are the spectral shape indices, where you basically take 3 bands. So this is like a fairly generic formula, but I will go into that a little bit more in the next slide to get a bit more of a geometric intuition, because those are very commonly used. The core idea here, and I take here the floating algae index as an example, uses 3 bands, and you basically measure the humpness of a spectrum. So you have like one band on the left, one band on the right, and one band on the center. And you do that both for here the interesting Anabaeana species, but also here for the water spectra, for instance. And then you basically interpolate between these 2 points, which are interpolated near-infrared reflectance. And then you can compare this interpolated near-infrared reflectance with the actual measured near-infrared reflectance, and that will be the floating algae index. So basically the real near-infrared reflectance versus the interpolated one. And you see that you basically measure the humpiness of the spectra here. So you take the left and the right, and you see how much does this hump deviate from the average between the two. Because when we compare with the water, basically the floating algae index will be close to 0, because there's no humpiness in this water spectrum. So this is also known among different names, for instance, the fluorescence line height in another paper here. So you see the same idea coming up with the left band and the right band. And here the difference in the interpolation is basically the humpiness of the spectra. So that's a fairly important concept if you see a spectra, how to design a spectral index, either with a normalized difference index or with a spectral shape index, to identify, to get a number out of that spectra, so you can perform a classification on that fairly easily. And here's an example where you can design a photocyanin indicator and a chlorophyll A indicator, basically by using spectral shape indices at specific bands that then need to be aligned with your sensor. So knowing the bands of your sensor is important, and knowing basically the chemistry of the photosynthesis in your algae is important. to then design spectral indices specifically for these use cases. Here's another example, same type of configuration and with the MERIS sensor, where you can design this photocyanine indicator and a chlorophyll A indicator. And basically by using your knowledge about the photosynthetic system in cyanobacteria and the sensor that you are working with. So let's look at some of these bands and see how relevant they are for like cyanobacteria in this case. So you see that MODIS has a few nice bands in the near infrared range, so you can measure the slope fairly well to get like a chlorophyll type of indicator. But Sentinel-3 has a few more in this range. For instance, he has 3 bands in this range, and also one on the photocyanine dip, but there's not a band on this like high reflectance area. So the satellites, they come with specific bands, and you need to know what is like useful for your particular use case. And also some of these reflectance bands are fairly well aligned with the absorbance of chlorophyll. So there has been a specific idea behind why these bands were put in these areas for you and for us to be able to design spectral indices in these cases. So as a takeaway of the role of Earth observation for harmful algal blooms, there are mainly 2 families of sensors. So there's the high resolution sensors with Sentinel-2 and Landsat, and low resolution sensors with MODIS, Sentinel-3 and so on. And they are basically serving 2 different purposes, one being like more large scale analysis, the other one being more on a regional scale with like the smaller pixels. And the other one is that harmful algal blooms can come in many shapes and forms and colors. So there's always a need for a custom case-by-case design of indices based on the algae of interest and the available sensor. So that's why it's important to go deep into the literature, see what other people have proposed for these specific cases to be able to distinguish different types of algae in the first place. And then there's like 2 types of spectral indices that you can use. So normalized different indices like NDVI, so to measure the steepness of the spectra, or the spectral shape indices like floating algae index to measure the humps or the troughs in the spectrum. And by designing these indices for specific bands, you should be able to create fairly reliable indicators for specific pigments or for specific algae blooms. And you can test this, and you can try this with a short exercise in designing a chlorophyll A index in Google Earth Engine. I've put the links also in the description on the TODO script, where you have to fill in the right bands, and then the solution script if you want to jump to the solution right away. The key point here is that we have here this algal bloom in the North Sea that is a bit visible in true color, but it's much more visible when you design a specific chlorophyll A index. And how to design this? I mean, you can look up the literature, but the key point here is basically to analyze the spectra that you have and with the absorbance of chlorophyll A, and then choose the bands accordingly that you're interested in, and that you think may be particularly helpful in identifying the chlorophyll A in the first place. So let's jump to the second part of marine litter detection, so the second field, which takes a lot of ideas from the floating algae field, but applies that to a different area of research and different types of materials. And I want to start with one particular use case study here in Durban, South Africa, where we had a fairly large precipitation event in 2019, where heavy rain caused like high floods in the Durban area, and together with mismanaged waste, the flood dislodged a large amount of litter into the Durban harbor with some social media pictures I will show in the next slide. But eventually it got washed out into the open Indian Ocean, where it's basically an environmental hazard, and it decomposes into microplastics and causes all kinds of problems. So here are some examples of this Durban harbor to show a bit the severity of this event, that there's a large amount of pollutants basically in the harbor that eventually made it out to the Indian Ocean. And when we look at the Sentinel-2 image a few days later, where you still see some of the clouds from the rain event, you see here on these like more annotated areas that you see large lines of outwash that in that case contain a lot of plastics and litter on the sea surface as they agglomerate into like larger patches. And measuring the origin and the destination of these litter patches is of course very interesting when you want to understand like what are the causes and what are the environmental effects of plastic in the environment from these types of events. And naturally it's a fairly large environmental problem with many million tons of plastic waste entering the marine environment. They're often deposited on beaches and they decompose and sink to the ocean floor, where they enter the health and the food chain of the animals, and eventually our food chain. And the main part here is that marine plastics, they decompose from the macro scale to the meso and the micro scale, where you have then, if you go one step further into the nanoscale, they start to enter really the biology of all living animals, and you will find macroplastics and microplastics basically everywhere on the planet at this point. So there's a large impact on microplastics on the aquatic life. So they're reducing food intake, delaying growth, causing damage, abnormal behavior. The detailed parts are not super understood yet, as it is a fairly active area of research, but you will be able to find micro and nanoplastics basically everywhere in the environment. So naturally, we are interested in using in situ studies and remote sensing to quantify and locate marine plastics at different scales. So at one side, you have in situ studies where you can perform like transect studies and you count how many amounts of litter did you find at specific areas. You can have UAV-based surveys, mainly to support in situ studies. Or you can also go on the larger scale on satellite Earth observation, where you detect agglomerations and generic marine debris that can contain marine litter, but at a larger, more agglomerated scale. So let's go to each of those. First, on the ground hands-on side, there's been on-site studies on river systems. So here you can deploy, for instance, a bucket of a river, and then you can count at the end how many plastic debris or litter debris were caught in this bucket. And if you do that systematically across the entire Rhine, you can make a fairly good picture of what is the composition and the quantity of different plastics caught in different areas. Here's a picture taken from the Rhine at Bonn, and you see that this ship here caught a lot of driftwood, and naturally in the driftwood you will find plastics floating on the surface. Detecting these at a large scale can be a task for remote sensing to be able to quantify the amount of plastics that enter the environment through the Rhine River. So naturally, there are citizen science-based surveys since many years. You may have been taking part of one of these already, where people go out and they collect litter on the beaches and then they quantify and they count how much they have found. So there's especially standardized protocols and classification systems that are good to follow, because you can then do an analysis of how did things change over time and how is the composition. And basically that works through standardized transect surveys, where you go 100 meters along and you search a certain width along the beach, and then you can quantify how many bottles at what size did you find, and then you have a fairly good map of the density. And that can lead to these types of studies where you have 20 years of beach plastic litter data, where you can really start to analyze how the change happened in the last 20 years at different beaches, where it was increasing, where it was decreasing, and what are then the causes of that, which is the subsequent question that you can try to answer once you know in what locations did it increase and where did it decrease. And here, naturally, we're going to the second part, remote sensing and Earth observation can help. On one side, on a UAV scale, to support these transect studies and those acquisition initiatives by taking pictures and then quantifying objects automatically. So naturally, a question is, how high should you fly your UAV to basically still be able to distinguish individual objects, and that the height of the UAV causes the pixel size to change. And then you can use computer vision to detect different types of objects individually, which is a really nice approach to automate this quantification system. So here is a computer vision pipeline where you have an image of different types of debris, and this particular model tries to split it up into two classification schemes. So one first being a bit more broad into is there litter or is there not litter, and water and vegetation. And that is then inputted again in this quantification part where both are convolutional neural networks, and then you can start to really try to distinguish different types of litter that are then interesting, on following these OSPA categorizations. So you can get an estimated number of total litter items and then an estimated number of different classes. And you can do this at larger scales here, for instance, in the Coastal Marine Litter Observatory, where you can upload your images from UAVs and they're automatically processed to get these types of litter density maps, where that really helps to scale up this UAV type of surveys through modern computer vision and machine learning approaches. So to summarize here a bit, we have the UAV side that is mainly there to support these on-ground transects. They have a high spatial resolution, so you can identify individual objects, but they're also relatively expensive to scale, at least when you want to go to a national or continental scale. They usually have a single image acquisition and often only RGB, which is not that bad because at this resolution you can mainly look at the geometry of objects to do object detection rather than spectral analysis. On a satellite side, you can have daily and weekly monitoring, and you have a good multispectral signal that will allow you to distinguish, in theory, different types of objects. But the low spatial resolution leads to a lot of mixed pixels, and you can only really work on agglomerations of objects within a pixel. But naturally, there's some agglomerations on the sea surface that help objects to move together into large patches, here like windrows and ocean fronts, as you can see here. And then these agglomerations are then large enough to fill like a substantial part of a pixel, and so that it's still detectable with a remote sensing sensor. And here, for instance, there's a specific type of agglomeration caused by windrows, where wind flows over the sea surface at a constant rate that causes these rotating barrel movements. And they cause these types of windrows that you can see on the surface. And if you then systematically fish along these windrows, you can collect quite a substantial amount of plastic. So most of these materials here are not plastic, it's more like natural and organic materials. But because these agglomerating factors are pushing things together, you will be able to collect a quite substantial amount of, like here, 80 kilogram of litter by 1 kilometer of length. But the challenge here, of course, is that there is a lot of things floating. We've seen harmful algal blooms before, but just because there's some kind of line visible doesn't necessarily imply there's plastic or litter. So it could also be like sargassum, plants or oil. Pumice is a very common floating rock after volcanoes. Pollen is also very seasonal cover on the surface, and sea snot is also a topic, which is like also agglomeration of different marine mucilage. But there have been quite a bit of studies on terms of artificial targets to study the spectral reflectance of different plastics on the water surface, as here, or slightly submerged, as down here. And there's basically ongoing research with deployed targets here with the Plastic Litter Project, and where you wait for satellites to overpass and analyze the spectrum response. So there's mainly this trade-off between spectral and spatial trade-off. So you have like Sentinel-2 with a multispectral signal at 10-meter resolution. With PlanetScope, you can go a little bit of higher resolution, but you only have 8 or 4 or 5 spectral bands depending on the type of sensor. So in general, it is more easy to detect something floating on the sea surface, and distinguishing the individual type is not that easy in terms of what type of litter. If it's like algae or litter, you can start to work with like some indices sensitive to chlorophyll, but it's still a challenge with those sensors we have available. But people have proposed specific indices, and you should be able to recognize this type of shape now, because we measure the humpiness on this specific spectra, which is like the floating debris index proposed by Biermann, which is inspired by the floating algae index. Again, it's the same idea of having a left band and a right band, taking the interpolation and seeing how humpy is this triangle here. And that is fairly helpful for visualization and analysis. So here is the satellite image of Sentinel-2 from this Durban example I gave you in the beginning, where you have the debris are not particularly visible in RGB, but when you calculate this FDI index, you will be able to see these debris still in the open water. And again, here I provided an interactive exercise with the TODO script and the solution script, and I'll put them also in the description below. With that, I will want to go to the conclusions, wrap up of takeaway and summary. We talked about those two main areas, like harmful algal blooms and marine litter debris. And harmful algal blooms is a fairly well-established research field, mainly driven by low spatial resolution satellites that are able to monitor large areas regularly of fairly well understood biophysical processes. We know, for instance, about the photocyanine and the cyanobacteria and the absorbance in the spectrum of chlorophyll. But we can take a lot of ideas from here in a more emergent research field like marine litter, marine debris monitoring, where current studies are ongoing of like how can we separate these different types of agglomerations with satellite data. What you should take away from this online lecture here is basically an understanding of spectral properties of algae and cyanobacteria, and especially the difference to land vegetation. Because photosynthesis, the water is not the same spectra as on the land, as we have, for instance, not the red edge. And the second one is the ability to assign spectral indices based on your knowledge about the spectral properties of the problem. With that, thank you very much for watching. I put links below to the exercises and the slides, and you're welcome to move forward and backwards in the slider to go to specific parts again.